Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello, welcome back to another episode of the A-Level Cookbook. Today we're going to be going through June 2021, Paper 2 from AQA A-Level Biology. Describe the process of glycolysis. Okay, so in photosynthesis and respiration, there are examples of metabolic pathways, which means you have a series of reactions controlled by enzymes. The way I personally recommend learning them is by learning them through big flowcharts, like so. So what happens in glycolysis is you first of all phosphorylate glucose into glucose phosphate, and that uses up an ATP. So you've got ATP, which is hydrolyzed into ADP, and the PI goes onto the glucose, like so. Then what happens is you do the same thing again and you get hexose, because glucose is a 6-carbon sugar, hexose, and then bisphosphate. It's got two of them, like that. And unsurprisingly, that also uses an ATP, which is hydrolyzed to ADP, and PI, which goes there. Then what happens is that this splits into two molecules of triose phosphate. You cannot just say TP, you need to say the whole thing, triose phosphate. You can abbreviate it later, but you need to say that. Now what happens to triose phosphate is that it is oxidized to pyruvate. When you oxidize something in biology, or anything in the universe to be honest, you need to reduce something else. So when triose phosphate is being reduced, two NADs are being reduced into two reduced NADs, like so. Not NADP, because we're not in plants, we're not doing photosynthesis, we're doing respiration. So that's this process here, and you should do that with all of the stages of respiration. It helps a lot just memorizing it and also understanding what's going on. So describe the process of glycolysis. In this answer you would say glucose is phosphorylated using ATP, and then you need to talk about what happens next. So you this when this forms glucose phosphate and then hexose bisphosphate and then it forms triose phosphate, and then what you do is you oxidize triose phosphate to pyruvate. At the same time, NAD is reduced into reduced NAD. Now it seems a bit silly to write that all the time, saying NAD is reduced to reduced NAD and reduced NAD is oxidized to NAD, but those are marked, so you should always say that. And as well as that, one thing I forgot to mention, sorry, is that there is when this process happens, where you've got triose phosphate and pyruvate, you actually regain ATP. So 4 ADP and inorganic phosphates form 4 ATP. But because you use 2 up at the start, here and here, you've got a net gain of 2. So there is a net gain of 2 ATP. And there you go, that's how you would do the first one. So malonate inhibits a reaction in the Krebs cycle. Explain why malonate would decrease the uptake of oxygen in a respiring cell. So the whole point of the Krebs cycle is to generate as much reduced FAD and reduced NAD as possible. And then what happens is that reduced NAD and FAD goes to oxidative phosphorylation. The electron is released as the reduced NAD and FAD get oxidized. The electron moves down an electron transport chain. You get the movement of protons across a concentration gradient, blah, 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 and you make ATP. But those electrons need to go somewhere. They don't just float around chilling everywhere. That's not how things work in life. So what happens is that they combine with oxygen and proton. The oxygen comes from the blood, and that's where you get water. So that explains why respiration's formula is glucose plus oxygen gives you carbon dioxide and water. So all of those stages that I described require reduced NAD or reduced FAD. So there are fewer reduced NAD FAD molecules, and oxygen is the final electron acceptor. Oxygen is the last thing that takes that electron after all the things it's been through. Explain how a resting potential is maintained across an axon membrane in a neuron. Okay, so I'm going to quickly describe how this works. So you've got your membrane here, whatever. And what you do is you've got your sodium-potassium pumps. So 3Na plus gets pumped out like this for every 2K plus in. However, there's also a nice little potassium ion channel like so, where potassium can just leak out freely, and that's how it maintains this electrochemical gradient. So even though you have positive ions moving in and out of the cell, in total there are more positive outside the cell than there is in. So that gives you that resting membrane potential, because you're shoving out sodium, you're taking in a bit of potassium, but the potassium's free to get out. So the sodium moves via active transport, so this exchange happens through active transport, but this movement here happens by facilitated diffusion. What happens is that sodium ions are actively transported out. You cannot just say sodium and potassium, you need to say sodium ions and potassium ions, because they are dissolved in solution and they're missing an electron, whereas sodium on its own has all its electrons and is a solid. So just make sure you say that. So sodium ions are actively transported out, and K plus ions are actively transported in and diffuse out. So they just freely move by facilitated diffusion out. And they need to describe sort of the end effect of all of this. So in general, the membrane is more permeable to potassium ions because they're moving in and they're free to move out, whereas sodium can only really get out. So the membrane is more permeable to potassium ions. Explain why the speed of transmission of impulses is faster along a myelinated neuron than a non-myelinated axon. And how this works is when you've got myelin, you've got, let's say this is your neuron, you've got these patches here of myelin, and you've got these bits of bare neuron between them called nodes of Ranvier. So depolarization happens at the nodes of Ranvier, but the myelin is an insulator. It doesn't let an electrical impulse go through it. So what happens is it has to jump over like this. So that's actually faster. It's called saltatory conduction, because if you have to go through the entire length of the neuron, the depolarization has to go through the whole thing, and that's bloody slow. Whereas if you bounce from the node to node, that gives you faster conduction. And there's a lot of disorders where they're called demyelinating disorders, where the myelin actually gets destroyed and the impulse has to travel through the whole thing, or you get scar tissue, so they get impeded like that as well. So in this question here, all you need to do is talk about that. So we would say that myelin is an electrical insulator, and then between myelin are areas with no myelin, known as nodes of Ranvier, where depolarization happens. The depolarization jumps from node to node, and this is called saltatory conduction. Saltatory just means in general like bouncing like that. There's a type of erosion in geography where rocks break down called saltatory erosion, and that's when the same thing happens. Okay, so a scientist investigated the effect of inhibitors on neurons. She added a respiratory inhibitor to a neuron. The resting potential of the neuron changed from minus 70 millivolts to 0 millivolts. Explain why. Okay, so normally, like we said, sodium is out and potassium is in, and that's maintained by by active transport using a sodium-potassium pump, which requires ATP. That keeps the outside more positive compared to the inside, which is more negative, which is why it's minus 70 millivolts. If you've impeded respiration, you've got less ATP, so there's less active transport, so that electrochemical gradient cannot be maintained. And that's exactly what your answer is here. So there is less ATP produced, therefore there's less active transport using the sodium-potassium pump. Therefore, the electrochemical, can no longer be maintained. You could also paraphrase and say something like, there's no net movement of sodium-potassium ions, or there's an equilibrium that's met. But that's how you would answer this. Figure 1 shows an investigation into growth factors in plants. So we've got the tip removed from a shoot of a plant, and then the tip replaced from one side of the cut shoot, and then growth curvature occurs without a directional light source. Use your knowledge of indole acetic acid, IAA, to explain the growth curvature shown in figure 1. So whenever it comes to questions like these, you need to identify whether it's a shoot or a root, because if you have a shoot, what happens is IAA promotes growth and elongation on the side it's on if it's in a higher concentration, whereas in roots it inhibits this. So in this case, this is a shoot, so in the shoot tip you're going to have IAA produced, which moves across to wherever growth is required, and then it causes elongation of cells more on one side than the other. So if you've got this here and I've got loads of IAA here, if this part elongates, then it's going to make it bend like that. So the tip produces IAA, and this diffuses into the shoot, and this causes cell elongation. Alright, this is crucial that you talk about cell elongation. It's not that they make more cells or anything, it is that elongation happens. A bioassay is a method to determine the concentration of a substance by its effect on living tissues. Figure 2 shows the practical procedure used in a growth curvature bioassay to determine the concentration of IAA in shoot tips. So we've got the shoot tips placed on agar. You remove the shoot tips and the agar is cut into smaller blocks. Each block is placed on one side of the cut shoot, and the shoot is kept in the dark and the degree of curvature is measured. Figure 3 shows the calibration curve for this growth curvature bioassay. So let's have a look at what's going on here. So we've got a degree of curvature on this axis, we've got IAA concentration on this agar block, so we can use this curve to figure out how much IAA would elicit how much curvature. If we have something with curvature and we measure it, we can work out how much IAA there probably was in that agar block. That's the whole point of calibration curves, is to be able to do that. Using the procedure in figure 2 and the calibration curve in figure 3, describe how you would compare the IAA concentration in shoot tips from two different plant species. In your answer, you should refer to all the variables that should be controlled to produce a valid comparison. Okay, so obviously you would get shoot tips from different species, place them on agar, cut them into smaller blocks, place it on the side of the cut shoot, and then record the curvature. And then what you could do is you would use the curvature here to work out the concentration. Obviously, as we can see here, higher curvature means higher IAA concentrations. So we need to think about everything that we need to control to do this. So in order to make this a valid comparison, we need to think about the factors that essentially will affect the concentration of IAA. So first of all, if we have a massive tip size, that's going to have loads more IAA production and amounts compared to a tiny one. So we need to control the tip size. As well as that, we should probably also do the shoot size as well, sorry. So shoot and tip size. They've removed shoot tips and agar has been cut into smaller blocks. So obviously we need to control how much agar we're using, the size of the agar block. Because if we have like a massive block and it's absorbed loads and loads of IAA, then that's going to obviously mess up our results. So we need to have the size of the agar block controlled as well. Now, we also need to think about what kind of plants we're getting these from. So obviously they're two different species, but what if we have like a really wilted, battered up plant that's gone through the trenches versus like a really fresh, nice plant? So we need to try and find shoots at the same stage of development or growth. So same stage of development. And then what did they do? They kept it in the dark. So obviously we need to control how long they're kept in the dark. So time period kept in the dark. What else can we do? Well, I mean, factors that affect growth. So there's temperature. Or what else would we do? We'd obviously repeat as well and calculate a mean in real life. And those are some pointers that I would say. So whenever you get questions where you're interpreting like a bioassay or anything that involves biology experiments, you need to think about the size, the volume, the shape, temperature, pH, what substances are using, how much of each substance are using, the organism, how old the organism is, how young the organism is, and things like that. Those are just general sort of pointers. In this case, it's saying to ignore pH, carbon dioxide, humidity, nutrients, light, and water, because we're under the assumption that they've been given enough of that. We're only talking about factors that could directly affect the IAA concentration and the effects that it can have on the end. So obviously IAA concentration is affected by the size of the tip, the size of the agar block, the stage of development, and then obviously the effects it has is controlled by how long it's kept in the dark and also its temperature. A scientist investigated the effect of a directional light stimulus on the distribution of IA and shoot tips. The scientists set up 3 experiments as shown in figure 4. All variables are controlled apart from the exposure to light. So we've got our agar block, we've got our shoot tip, and we've got the light here. This is in the dark. And then here we've got an impermeable barrier between A and B, and we've got a direction of light here. So let's see what's going on. So she then used the growth curvature bioassay to compare the IA concentrations in the agar blocks between experiment 1, 2, 3, and 3B. So we got the degree of curvature here. Now we need to relate this back to the graph that they've given us before. The higher degree of curvature implies that there's a higher concentration of IAA in the block because they're proportional. There's a nice straight line there. So that's what they're trying to catch you out on here. So now it says, state 2 conclusions about IAA that you can make from the results shown in the table. So in experiment 1, the degree of curvature was 17.69, which means there's quite a decent amount of IAA. Whereas in experiment 2, you know, it's not very much different. So it doesn't. So maybe light and dark doesn't really have any sort of impact on the IAA. So we can think about that. And then 3A it says 11.22, and then 3B is 6.50. So what that's implying is that IAA is lower here and higher here. Obviously, the first conclusion we can draw is that light doesn't really have an effect on IAA production. So light does not have an effect on IAA production. So we figured that part out. Now we need to think about the direction of light in the barrier sort of element here. So 3A's got loads of IAA, 3B's got very little IAA. So it looks like that IAA moves away from light in this case. In fruit flies, males have the sex chromosomes XY and the females have XX, so similar to human males and females. In fruit flies, a gene for eye color is carried on the X chromosome, and the allele for red eyes, big R, is dominant for the little r, white eyes. So I'm going to write that here just to remind me. And these are X-linked. So it says male fruit flies are more likely to have, more likely than female fruit flies to have white eyes. Explain why. So if we look at the X and Y chromosomes, so XX and XY. The X chromosomes are like nice and lengthy, beautiful chromosomes, and then Y is this like absolutely battered piece of rubbish, like utter, like, you know, it's missing all of this, bro. Like, what's going on here? So if you have a recessive allele here, there's nothing here to counteract it. So there's no dominant that might outdo its expression. So whatever this is will be expressed. Whereas if you have a recessive allele here, chances are on the other chromosome you're gonna have a dominant allele here. So it's less likely for a female with XX to express that. So it says male fruit flies are more likely to express it. Explain why. Well, you know, the females would need two recessive alleles, whereas males only have one. Males only have one X chromosome, so they only need one. Have one X chromosome. Therefore, they only need one copy of a recessive allele. A female fruit fly with white eyes was crossed with a male fruit fly with red eyes to produce a large number of offspring. Tick one box next to the statement which correctly describes the phenotypes produced from this cross. So you can just quickly do the cross, it won't hurt you. So a female fruit fly with white eyes, so that's going to be X little r, and then X big R, Y. So X big R, X little r, X big R, X little r, X little r, Y, X little r, Y. So you can see that all the females are red-eyed and all the males are white-eyed. So where's that? All females red-eyed, all males white-eyed. There you go, bang. In fruit flies, the gene for body color and for wing development are not on the sex chromosomes. The allele for gray body color G is dominant to black, so big G is gray, little g is black. The allele for long wings, big L, is dominant and little l is recessive. So that's a geneticist carried out a cross between fruit flies with grey bodies and long wings, heterozygous for both genes, and fruit flies with black bodies and short wings, and here are the results. So what they did is they crossed big G, big L, little l with little g, little l, little l. And it says explain the results in table 2. Okay, so we need to look at the phenotypes and relate them back to the genotypes. So they've got a grey body and long wings. So that means if it's grey body, it's got to be big G, little g, and then long wings, it has to be big L, little l. And it says black body and short wings, so that's got to be little g, little l, little l. And those phenotypes match the parents quite quickly, quite considerably, and also the number of offspring aren't massively different. So what's probably going on here is you've got autosomal linkage. It's not following sort of a standard pattern. So here you would say there's autosomal linkage, and we need to explain why that's the case. So if you think about what's going on here, then you've got no offspring with these phenotypes. So big G, little l, and little g, big L, little l. So you've got no black body long wings, and you've got no grey body short wings. So that implies that there is autosomal linkage. Now we need to talk about what that means. So when you have autosomal linkage, what that means is that you've got two genes on the same chromosomes. What goes on is that they're on the same chromosomes. So imagine we've got a pair of homologous chromosomes here. It's mum and dad's. Let's say these are the genes here. They're quite close together. So when you've got crossing over that happens like this, like, they're so close together that they don't actually get passed on, and they just kind of stay in that same chromosome, like so. Whereas let's say there was another one down here for something else. That one's been exchanged off, so it comes onto there. So that's what's happening here, is that they're not being crossed over. So they stay close. They stay together. So they are not crossed over. So that's what autosomal linkage is. It's where they're so close together that they actually stay together during independent segregation and crossing over. So those alleles get passed on together to the offspring, which is why you get these sort of things here where you don't have any any of these middle phenotypes. Even though if you do a cross, you would expect those to come up. If you cross these, you would expect these to come up, but they aren't present here. Okay, so the first generation of a population of fruit flies had 50 females. Calculate how many female fruit flies will be produced in this population in the fifth generation. You can assume each female produces 400 offspring each generation. Half of each offspring produced in the generation is female. There is no immigration or emigration. No flies die before reproducing. And show your working and give your answer in standard form. Okay, so we've got 50 females, and each one produces 400 offspring. So we've got 400 offspring. So this is the first generation. So 50 times 400 gives you 20,000. Now of those 20,000, half are female, so we need to divide by 2. And that gives you 10,000. And that's your second generation of females. Now again, each one of them has 400, so times by 400. And you've got 4 times 10 to the 6. But again, remember, half of them are female, so divide by 2. And that gives you 2 times 10 to the 6. That's your generation 3, and then you rinse and repeat the cycle. So I'm going to times it by 400, divide by 2, and that gives me 4 times 10 to the 8 as generation 4, then times 400, divide by 2, and that gives me the last generation, which is 8 times 10 to the 10. Neonatal diabetes is a disease that affects newly born children. This disease is caused by a change in the amino acid sequence of insulin. This change prevents insulin from binding to its receptor. Explain why this change prevents insulin binding to its receptor. So insulin is a hormone, and hormones are proteins. So if it's a protein and you're changing the amino acid sequence, you're changing the primary structure, the secondary structure, the tertiary structure. Because you're changing its tertiary structure, has different hydrogen and ionic bonds, so therefore it has a different 3D shape, and it's no longer complementary to the insulin receptor. We are not talking about enzymes here. There's no active site. That's not how this works. So I see a lot of people, what they'll do is they'll just write a half-assed answer about enzyme active sites when this is not an enzyme. Alright? So it stops insulin binding to its receptor. Insulin binds to the receptor and it does its job. So what happens is you've got a changed tertiary structure. Now what does that mean? It's no longer complementary to its receptor. It doesn't form enzyme substrate complexes. It doesn't, have an active site. And if you write that, you will not get the marks. Also, it's talking about insulin, not the receptor, so you don't get any marks if you say anything regarding the structure of the receptor. Okay, so phosphoinositide 3 kinase, PI3K, is an enzyme in several metabolic processes, and Figure 5 shows the role of PI3K in the control of blood glucose concentration. So insulin binds to the insulin receptor, IRS activates PI3K, which activates AKT, AKT stimulates movement of vesicles, and what happens is you've got these vesicles that implant glucose channel proteins, and then you've got glucose coming in like that. So decreasing the activity of PI3K can cause type 2 diabetes. Explain why. So you need to look at sort of the chain that's going on here and the pathways and see what problem's going on. Decreased activity of PI3K means you've got less AKT activated, so less AKT activation. If you've got less AKT activation, that means fewer vesicles with glucose channel proteins. Move to the surface membrane. Therefore, you've got less diffusion of glucose into the cell, because that's what insulin normally does. Therefore, increased blood glucose concentration. And that is your diabetes. The reason it's not type 1 diabetes is because type 1 diabetes means there's an absence of insulin production. There's no insulin made to start with. Insulin's still here and produced and trying to do its job, it just doesn't work. And that's what type 2 diabetes is where you've got insulin still being made, but it just doesn't work. Okay, so using your knowledge of the kidney, explain why glucose is found in the urine of a person with untreated diabetes. So just as a quick reminder, you've got your Bowman's capsule and your glomerulus and all of that kind of stuff. You've got your proximal convoluted tubule, your loop of Henry, blah, blah, blah. Henle, sorry. And then you've got your distal convoluted tubule and your collecting duct. So what happens is you've got ultrafiltration happening here, and then along the length of this, you have selective reabsorption. So you squeeze out absolutely everything except big proteins, and like blood. So you're including glucose. It goes into the Bowman's capsule, down the proximal convoluted tubule, blah blah blah. But what happens is that you have reabsorption of glucose at all of these points using carrier and co-transport proteins, right? Even if it's against the concentration gradient, like it will still your kidneys try to take up as much glucose as they possibly can. But because this requires the use of carrier or co-transport proteins, you only have a limited number of them. So if you've got such a massive amount of glucose coming in here and like chilling, it's not going to be able to be taken up. So you actually pee it out because it gets to the end of this pathway in the collecting ducts, to the ureter, to the bladder, and you pee it out. So that's why that happens. You've maxed out all your carrier co-transport proteins. So you have a high blood glucose concentration in the glomerular filtrate, or blood, or filtrate, allowance any of them. So that means not all glucose is reabsorbed. You need to know where it's reabsorbed, and that's reabsorbed at the proximal convoluted tubule. So not all glucose is reabsorbed at the, I'm going to write PCT for short, but you write the whole thing. Because, you need to say why, because it says explain. It's because all co-transport or carrier proteins are saturated. They're all being used up at maximum capacity. Like, there's no extra way of getting it in. Myelodysplastic syndromes, MDS, are a group of malignant cancers. So in MDS, the bone marrow does not produce healthy bone cells. Hematopoietic stem cell transplantation is one of the treatments for MDS. In HSCT, the patient receives stem cells from the bone marrow of a person who does not have MDS. Before the treatment starts, the patient's faulty bone marrow is destroyed. For some patients, HSCT is an effective treatment for MDS, and explain how. So it's told you here that in MDS, you do not produce healthy blood cells. So if you replace the stem cells with stem cells that aren't MDS stem cells, they're going to make healthy blood. So, transplanted stem cells divide and specialize. And this produces healthy blood cells. Since they've destroyed the previous bone marrow, there's no MDS cells. Well, there you go. MDS can develop from epigenetic changes to tumor suppressor genes. In some patients, the drug I'm familiar with seeing as azathioprine, so whoops. The drug AZA reduces the effect of MDS. AZA is an inhibitor of DNA methyltransferases. These enzymes add methyl groups to cytosine bases. Suggest and explain how AZA can reduce the effects of MDS in some patients. Epigenetic control involves methylation and acetylation. So when you're methylating a gene, you're adding a CH3 on, right? If you're adding a CH3 on, it's harder for RNA polymerase to do its job. So RNA polymerase cannot make an mRNA copy as easily, so it's not transcripted. Therefore, there's no translation. You're not making the protein. Whereas if you have so this is increased methylation. If you've got increased acetylation, what this does is it makes the histone proteins around DNA sort of unwind and make it a nice and easy for RNA polymerase to do its job. So that's how it generally works. So in this case, it's saying that it inhibits DNA methyltransferases, and these enzymes add methyl groups to cytosine bases. So what's happening is that it's reducing, AZA that is reducing methylation of a tumor suppressor gene. So I'm going to write TSG for short, right? So this means that more of, the TSG is now expressed. And then you need to talk about what that involves. So it produces proteins that inhibit mitosis. Therefore, there's less uncontrolled cell division. So you need to mention uncontrolled cell division and mitosis because this is a cancer. So whenever you get questions on tumor suppressor genes or proto/oncogenes, you need to talk about uncontrolled cell division, mitosis, and the production of a tumor. So in this case, that's what we've talked about here. Scientists investigated the effectiveness of AZA in patients with MDS. A total of 360 patients were randomized in a ratio of 1:1 to reduce AZA or conventional drugs control, and here's the results. So those with AZA so what's going on here? So time after treatment and the proportion surviving. So obviously as time goes on, unfortunately, with the proportion surviving, the survival drops. Whereas AZA, you can see that more kind of survive overall here as they're dropping less. So for example, if we take 10 years, the survival is about 0.558. Whereas here, this is 0.7 something. So more of them are surviving taking AZA. Now, the control patients were treated with conventional drugs. Give 2 reasons why. Well, first of all, it's so that we can compare the effects of AZA. You cannot just say to allow comparison. That means absolutely nothing, because it could mean that you're comparing, God knows what, basically. You could be comparing the patient's characteristics, everything. So you need to say that you're comparing the effect of AZA specifically. You want to know if AZA is doing something or not. Not just allow comparison. That's never a mark, so don't ever just say that. The other thing is it says, why are they treated with conventional drugs? Ideally, wouldn't you want to have no drugs at all versus AZA? Well, the problem is that it's unethical because these people have a type of cancer, and if you're not giving them cancer therapy, that's kind of messed up. So it's unethical not to give conventional treatment. Because then you're just, leaving them with untreated cancer, and that's just not ethical. So yeah. Use figure 6 and the information provided to calculate the difference in the number of patients surviving at 10 months after treatment of AZA compared with conventional drugs. So at 10 months, the proportion that we're taking conventional drugs is 2, 4, 6, 8. So that's going to be 0.58. And then the proportion taking AZA is going to be 7, 2, 4. So 0.74. Okay, so this is just the proportion now. This isn't the whole number. So they've got a total of 360 patients, but they're split into 2 groups of 1:1. So each of these groups, have 180 in total. So we need to work out the proportion of 180 for each of them. So 0.58 times 180 gives you 104.4, and then 0.74 times 180 gives you 133.2. And then you work out the difference. The difference is just those two subtracted, which is 133.2 minus 104.4, which gives you 28.8. But you can't have 0.8 of a person, so that's 29. There you go. Again, these questions only 2 marks. Just don't bother if you can't do them. No point wasting your time. Just write something and then move on. Hepatitis B is a life-threatening liver infection caused by the hepatitis B virus HBV, and we've got our structure here of HBV. So we've got polymerates, we've got DNA, we've got lipid layer, capsid, HBsAg, attachment proteins. So HBsAg infects a liver cell. A liver cell is 25 micrometers in diameter. Use figure 7 to calculate how many times larger in diameter this cell is than HBV, and you should use the lipid layer to measure the diameter of HBV. Right, okay, I can't really do this because I'm doing this digitally, and I did try and it just didn't work out properly. So basically, I'm just going to quickly talk through what you do. So all you do is you measure this to this side with a ruler, the lipid layer, and then you compare using the scale here. So you need to use image equals actual times magnification on here. So you measure this, and that's your image, and your actual here, well, they've told you that's 10 nanometers. Make sure the units are the same and rearrange for magnification. Then what you do is you measure this line here that you got before, and then divide by the magnification, again using I equals AM. And then you've got you can rearrange this to get the actual size. Once you have the actual size, make sure the units are the same and then divide the two and you'll get your answer. Okay, so scientists investigated the effectiveness using two types of RNA interference molecules in reducing HBV replication, and they are short hairpin RNA, shRNA, and long hairpin RNA, so LhRNA. A scientist infected mouse liver cells with HBV and transferred either shRNA or LhRNA in these cells. They then determined the concentration of the attachment proteins HBsAg in these cells. The concentration of HBsAg is a measure of HBV replication. Right, so the data represented here represents plus or minus 2 standard deviations from the mean, which includes over 95% of the data. So the mean HBsAg concentration of the control, it's a percentage of the control. So you can see that there's a lot less here compared to the controls and a lot more here compared to the controls. So one method of transferring RNAi molecules into these cells involves combining these with a lipid. So just like this increases the uptake of RNAi molecules into cells. So remember that lipids could freely move across phospholipid bilayers. So the cells have a phospholipid bilayer. The cells have a phospholipid bilayer. So there's no need, for facilitated diffusion, so there's no need for a channel, a carrier protein or channel protein. So they're taken up very easily. Using all the information provided, evaluate the use of the two types of RNAi in treating hepatitis B in humans. Do not refer in your answer to how RNAi reduces HBV replication. So they're just talking about only using the data. So again, this is an evaluate thing, so you need to say what's for and what's against. So just to interpret this data, again, this is a mean HBsAg concentration percentage of control. So if this didn't work at all, like absolutely did not work, then these would be 100% because it would be a percentage of the control. So both of these at least do some work because this is 50% of the control and this is whatever that is when you read the scale. So already off the bat you can say that both more effective than a control. Now we need to look at everything else that's going on here. So we've got the standard deviations. The standard deviations do not overlap. Now if you've watched my videos, you'll know that I go absolutely apeshit when somebody says, oh, therefore the results are significant. It doesn't mean shit. You need to say what the results means. So the standard deviations do not overlap isn't a mark. You need to say the whole thing. So, well, sorry, it is a mark, but you need to say the whole thing. Please do it. So standard deviations do not overlap. Therefore, the differences in the means, or the differences in the results in the means, are not due to chance. Please just say that. Do not say the results are due to chance. You'll get 0 marks. I can guarantee it. Now, obviously, we need to talk about the problems of this. If we want hepatitis treatment, we want all of it to be gone, not just reduced. So HBsAg is not 0 here, so it doesn't get rid of all of it. So HBsAg, not 0. So it's not, going to get rid of everything. As well as that, are we testing it in humans? Are we testing it in petri dishes? Are we testing it in animals? You can see they've only tested in mice in this question. So only tested in mice. As well as that, we don't know any sample size. We don't know how long it's been going on. We don't know any numbers either. LhRNA is better than shRNA as well. So there's a bit of a difference between the two there. Describe and explain how the polymerase chain reaction is used to amplify a DNA fragment. So, oh man. Okay, basically all you do is you put the DNA fragment, so the DNA fragment, DNA polymerase, nucleotides, and primer, and you put them in a nice little mixture. You heat to 95 degrees Celsius. to break the hydrogen bonds. And then you reduce the temperature to like 40 something degrees to allow the primers to bind to the DNA strands, because the whole point of breaking those hydrogen bonds is to separate the DNA strands. Then you increase the temperature so DNA polymerase joins the nucleotides. Okay, so figure 9 shows the number of DNA molecules produced making a PCR. Explain the shape of the curve. So you've got a number of DNA here, and you've got a number of PCR cycles. You can see it's kind of low, and then bang, it shoots up, and then it levels off. So you need to think about what happens. In PCR, you're doubling each time. 2, 4, 8, 16, 32, 64, 128, and so on, and so on, and so on. So obviously that explains why it's a little bit slow, and then bang, it shoots up. But we need to think about why it levels off. Why would it level off? Well, there's no more nucleotides or primers left. So it doubles each cycle. So you get a exponential increase. Exponential just means something to the x. In this case, it would be 2 to the x. Exponential. It's the exponent of 2. And then as well as that, it levels off because there are no more nucleotides or primers. You know you've run out. So you can't really do it anymore. A coral reef is an underwater ecosystem formed by a ridge of mainly calcium carbonate deposits. Algae are photosynthesizing organisms. Some algae grow on coral reefs. Succession results in a wide variety of fish living on coral reefs. Describe a method that could be used to determine the mean percentage cover of algae on a coral reef. Do not include information on the difficulties of using your method underwater. Okay, so if we're looking at mean percentage cover, we are counting up how much, algae we can find in this area. So we're going to be using quadrats here. Now, whenever you're talking about the use of quadrats, you need to talk about how you're going to eliminate bias. So what that means is, let's say I just divide it into a nice old grid, and I'm like, oh yeah, that spot looks quite good, I'm going to put it there. That's bias. So what you want to do is you want to use a randomly generated number, so a random number generator, sorry, and then use that to give you coordinates. So what this means is, let's say we've got like A, B, 1, 2. Rather than me picking A1, which might look good, the random number generator might tell me to do B2 instead. So that means that I avoid getting that bias in. So in this question, what you need to do is you need to divide the area into coordinates and use a random number generator to generate coordinates to choose. To sample, sorry, I should say. And what you do is you use a large number of quadrats, because you don't want to use like 3, you need to use a good amount. And then obviously you count up how much, you count up the population of algae, and then you divide the total percentage by the number of the quadrats that you have. Because we want the mean percentage cover of all of it, so divide the total percentage of cover by the number of quadrats. And that'll give you that. Explain how succession results in a wide variety of fish living on coral reefs. Do not describe the process of succession in your answer. So all this question wants you to do is describe the end results of it. So if you've got a wide variety of species of fish, different fish eat different things, they live in different places. So that means you've got an increased variety of populations. You've got different, populations, different species and stuff, so of different species. So therefore, you've got an increase in habitats, different habitats, and niches. Because if you think about it, if you've got like a bajillion, it doesn't matter about you need to say different habitats, because you could have like 8 million of the same habitat, the same place that a fish lives in. So you've got 8 million of that same fish. If you've got 8 million different ones, you've got 8 million different fish. So it's just making sure you get that point across that you've got differences. It's the same with like food. You can't just say more food, you need to say more food sources, because different fish eat different things. If I've got 8 million of one type of thing a fish eats, then I've got 8 million of that one fish. Whereas if I've got 8 million different food sources, that's probably going to be like, a lot of different fish species. Ecologists investigated the effect of 2 fish species, the red band parrotfish and the ocean surgeonfish, on algal growth on an artificial reef. They made this artificial reef by submerging many large concrete blocks at a depth of 16 to 18 meters off the coast of Florida. They attached 4 large wire cages A, B, C, and D to each block and populated the cages as shown. So cage A has no fish, B has 2 red band parrotfish, C has 2 ocean surgeonfish, and D has 1 red band parrotfish and 1 ocean surgeonfish. Bloody hell. After 34 weeks, the ecologists measured the mean percentage cover of all algae within each set of wire cages. The ecologists used a standard test. The ecologists used the statistical test to find out whether the mean for each of the cages was significantly lower for the mean. Table 3 shows the probability p-values that the ecologists obtained using the statistical test. So it says, using all the information, evaluate the effect of the 2 species on algal growth on coral reefs. So what they're measuring here is they're measuring the mean for each cage is significantly lower than the mean for set A. So here, this is higher than 0.05, this is lower than 0.05, and this is also higher than 0.05. So therefore, C has a significant decrease. You can't say the results are significant. Please, you cannot say that. You need to say what they're talking about. So they're talking about the decrease compared to A. So C has a significant decrease compared to A because its p-value is less than 0.05. Therefore, therefore the difference is not due to chance. Almost caught myself out there. Oh my god. Not due to chance. Whereas in B and D, it's greater than 0.05. Therefore, the decrease is likely to be due to chance. Now, that only gives you about 3 marks. We need to talk about the rest. So they're talking about the effect of the 2 species on algal growth on coral reefs. Well, first of all, they've only done it on the coast of Florida, so they've not done it on other reefs, so we don't know if it's different, and we also don't know what species of algae that's used as well. It's not only talking about the statistical test, it's saying using all of the information. So make sure you read the whole question, and make sure you use everything they give you. Okay, right. So the amount of times you see a passage question and people just do not read it and they try and do the line stuff. If you watched every video I've ever talked about, I always complain about this. Bloody bro, just skim through it. Just do it. Unless you're really struggling for time, do it, because it just makes things a lot easier. It makes your life a lot easier. Right, I've already kind of read this. It's fine. Lake Malawi in East Africa has more species of fish than any other lake in the world. Suggest and explain how this speciation might have occurred. So let's read what it says. Lake Malawi in East Africa has more species of any other lake in the world. Oh my god, that's what line 1 says. Shit, man, I don't know what to say. Read around the bloody line, okay? The amount of times that I've had to help with questions like this, and they just read that one line and they're like, I can't find the answer. It's like, come on, bro. Many of these species have evolved from a common ancestor. Oh, we might be onto something now. Oh my god. Lake Malawi is one of the largest lakes in the world and was formed several million years ago. Since then, the water level has fluctuated greatly, and as a result, what is now a large lake was at one time many smaller, separate lakes. Yo, what? Huh? Maybe that might be a clue. Obviously, we need to decide what type of speciation this is. Is it allopatric or is it sympatric? So it's telling you that you had lots of different lakes beforehand if you read just a little bit more than the first bloody line, you know? So obviously we've got some geographical isolation here. So this is allopatric speciation because they are happening in 2 different populations away from each other rather than sympatric, which happens in the same. So what happens is that you've got these smaller separate lakes, and what they do is they have different environmental conditions. Because they have different environmental conditions, there's different selection pressures. Now, mutations lead to variation, which may give rise to an allele that increases the chance of survival and reproduction. Okay, so with these kinds of questions where it's natural selection, you're talking about the fact that mutations give rise to an allele that may give you an increased chance of reproduction and surviving and reproducing. So this passes on the allele, leading to an increased allele frequency over time, because you know the ones that don't have the allele are less likely to survive and reproduce. So you've got more of this one allele here. And what happens is that you've got geographical isolation, so there's no gene flow because they can't reproduce with each other, so there's no gene flow between the two. And eventually what happens is that they become different species and they cannot interbreed to provide fertile offspring. The percentage of forest cover in Malawi decreased between December 1990 and December 2016. Calculate the mean loss of forest cover in kilometers per squared per week during this time period. So let's have a look. So the country of Malawi has a total area of 118,000 kilometers squared. The actual land area is about 94,080 kilometers squared. So they've told you that forest covered 41% of the actual land area. So we're going to be using this number, not this number. So 1990 and then 2016. So we need to work out the actual values. So you would do 0.414 times that and then 0.264. So 0.414 times 94,080 gives you 38,949.12, and then you do the same for the 2016, which is going to be 26.4, so 0.264, and that gives you 24837.12. So now we need to work out the difference between the two of these, because that'll be the loss of forest stuff. So that's going to be 38,949.12 minus the answer, which gives you 14112. So now we've got the loss in kilometers squared, but they want it per week. So we need to work out how many weeks have actually elapsed between these two. So the number of years is going to be 2016 minus 1990. So 2016 minus 1990 is 26 years, which is 9490 days, because you do that times 365, and you divide it by 7, that'll give you how many weeks you've got, which is 1355.7. Therefore, the loss in kilometers squared per week is going to be 14112 divided by the answer you just got here, which is going to be 14112 divided by that, giving you 10.4. And there you go. Again, if you can't do this, don't bother wasting more than 2 minutes. So loss of nutrients into Lake Malawi has resulted in a decrease in sunfish populations. Line 12 to 13, explain why. So deforestation and farming along the shores of Lake Malawi have caused increased soil erosion and loss of nutrients into the lake. This has resulted in a decrease in sunfish populations. The mark release recapture method, okay fine, so we're actually stuck here because it doesn't tell us the answer. Oh no! So we need to think about what happens. So we've got nutrients leaching into a lake. What that means is that algae are going to start, developing and growing more and more and more. And what happens is you get eutrophication. They block light, so there's reduced photosynthesis in the water itself. Photosynthesis uses up carbon dioxide and releases oxygen. And obviously, the plants are now dead, so saprobionts, like microorganisms, are going to aerobically respire using up that oxygen, and there's less oxygen for fish to use to respire, and then boom, they are dead as well. So increased algal growth blocks light. Photosynthesizing. Plants die. Saprobionts aerobically respire. You need to clarify that it's aerobic respiration, or else you're not including the fact that they're using up the oxygen. And therefore there's less oxygen for fish to respire. Therefore, they die. Okay, so the mark release recapture method can be used to estimate the total size of a fish population. Explain how. So what have they said here? The mark release recapture method can be used to estimate the size of a fish in a population. However, this method can produce unreliable results in very large lakes. Okay, so let's have a look. So obviously we need to describe how this works. So first of all, we need to, you need to collect. Whoops, it's lagging. Collect fish and mark them in a way that isn't harmful. Or affects their survival. So you don't want to make them more obvious to predators. And then you need to allow them to evenly distribute after releasing. And then you recapture and count the marked ones. Recapture and count marked ones. And then you use the formula population equals number in first sample times the second over the marked fish in second, and that lets you estimate the population size. The reason you need them to have time to evenly distribute after releasing is because they need to divide themselves evenly. So imagine these dots are your population of just like everything, your entire ecosystem, and these are the recaptured fish. If you don't allow them to redistribute and they all sort of cluster here and you capture all of them again, that's going to make this higher, so the population's going to be smaller. Whereas if you spend too long and they bloody go like, you know, to the other corners of the entire planet and they voyage the 7 seas and like encounter pirates on the way, blah blah blah, then you're going to recapture like one of them and it's going to make the population like unreasonably massive. So you need to sort of balance it out and have a way of, you know, balancing it. So just why the mark release recapture method can produce unreliable results in very large lakes. Well, I've just said that, haven't I? So you're less likely to recapture marked fish. And therefore that makes this lower, so the population would go higher. Thank you very much for tuning in to today's episode of the A-Level Cookbook. I hope that helped. I'm trying to do as many of these as I can in the run-up to the exams. Sorry if they're a bit slow. Again, if this helps, please like, share, and subscribe. It goes a long way. I'm quite thankful for all the people that have been watching my stuff and commenting the really positive stuff. Like, it's really been a massive motivator. So thank you very much, genuinely, just from me personally, that you guys have been watching my stuff and finding it helpful. It's one of the most heartwarming feelings. So yeah, thank you very much.